everybody to our AIHI seminar series and the latecomers. And pleased to introduce Dr. Tony Badrick, who's the Chief Executive of the Royal College of Pathologists of Australasia Quality Assurance Programs. Tony's the President, or was the President, of the Australian Association of Clinical Biochemists from 2003 to 2007. He's also the Chair of the Faculty of Science of the Royal College of Pathologists. And he also has a few claims to fame, but the one that I'm pretty impressed with is that he's got 2 PhDs, one in quality management and one in pure maths. So that's for all of us, all the struggling PhD students. This will give you inspiration. So welcome, Tony.
Thank you. Thank you, Andrew. Thank you for inviting me, and thank you for coming. So I'm going to talk about pathology database mining. I'm not an expert. My background is I'm a clinical biochemist, so my interest is more in the application than the actual mining techniques themselves. But and also, consequently, there's a fair amount of detail on pathological things, but I don't really want you to pick up on that. It's really how the tools can be used. So I always like to start with a challenge. What's that? Sorry? No. Sort of, well, very indirectly. What can you see? Can you see anything?
A turtle climbing a tree.
No, but this is where you need a psychologist in the room. I'll give you a clue. There's an eye. There's another eye. There's a set of nostrils. There's 2 ears, so it's a cow. Okay. So there was a purpose. There was a purpose to that, and the purpose is it's like data mining big databases. Unless you know what you're looking for, you won't find it. So in terms of database articles, that's going through the roof because we've got the techniques now to do it, and we've got the databases that are out there to mine. And mining pathology data is big business, and that's because there is so much data out there. There's something like 4 million Australians have at least one pathology test done every year, and most of those pathology tests will consist of 20 or thereabouts tests. So, and most pathology laboratories will have data going back over 20 years. So there's an enormous repository of data there to mine. I'll just go through some very general aspects first, quickly, just to give you some idea of the uses of data mining, and then I'll hone down to more specifics, the stuff that I'm interested in. So you can use it to detect outliers, association, dependency modelling, where you're looking for relationships between variables. This is really sort of fundamental research, where you're trying to see if there are relationships between different analytes. And one of my colleagues who actually does a lot of the mining, his interest in this is that it's an alternative to using animal models. So using humans, and really every sort of disease that we want to find in a human's got. So somewhere out there, there's data that a human that we're trying to investigate a disease in has got the disease. We don't need to find a rat and create an artificial disease. The diseases are all there. We can use it for clustering, to discovering groups and structures, classification, regression, summarisation, okay? So this is just high-level stuff. There's lots of uses for data mining. And this is a very useful paper, I think, which sort of describes the uniqueness of the medical data, the problems that we have with medical data. I was at a conference last week and someone said that diagnosis is the most complex human activity there is. Rocket science is easy, it follows all those equations that Newton worked out 300 years ago. Nobody knows how to do diagnosis effectively. And I like to think about diagnosis this way. I think in maybe 10 or 20 years, medical diagnosis will become as accurate as weather forecasting is now. That's the hope. The problem with, and when you think about it, if you say that in terms of the weather, if you say tomorrow is going to be the same as today, there's about a 70% chance you're right. Yeah, well, Yeah. No, well, it's Friday evening in Melbourne. It's the same tomorrow as today. So there you go. So in terms of the sort of uniqueness of the medical data, that's because of the heterogeneity of medical data, ethical, legal issues about privacy, confidentiality, the statistical philosophy of doing mining, any sort of data mining. And I'll keep coming back to the stats because it's not all sort of black magic. There is sort of structures that you need to apply, and the special status of medicine. And that is, what's ethical? So the heterogeneity of medical data, it's because of the volume and complexity of medical data, MRIs, liver function tests, the genomic data that we're about to get, the whole world's, all the computers on Earth are going to be full of our genomes. Physicians' interpretation of, what's wrong with you? What is it? Fever, heat, febrile, high temperature? Sensitivity and specificity, it's really hard to define diseases, really hard to define in individuals because of the variation what a disease is. Sensitivity is poor, specificity is even worse. Who knows what sort of distribution and a lot of this data follows, and there are problems with the way that we describe this disease. These are just to reinforce the sort of the heterogeneity of the synonyms for the same disease. All these represent the same thing. Ethical issues, who owns the data? Patients? Pathology labs? The pathology labs have to keep data, particularly genomic data now, for 100 years. I don't even know how that's going to work. Who owns it 100 years down the track? Somebody's great-grandson? Legal issues with disclosure of that data, privacy. Who's going to administer this data for hundreds of years? And is it valuable if you use my specific disease to discover something? Shouldn't I get a cut if there's money in it? The statistical problems, there's an ambush in statistics. You start looking for something and you find something else. You start treating a patient, so patients have a particular disease, you start treating them. If you're doing a sort of a randomised controlled trial, and you give one lot of patients the drug and the other lot not the drug, and the lot with the drug get better, are you going to not give the drug to the other group? Of course you are. So in the middle of all these elaborate trials, you change tack because you can't disadvantage patients. Data mining is a superset of statistics. It's still got a statistical base. And it's a knowledge discovery process. And I'll show you what I think is a great example of that later on. Medical data, there's unique technical issues. As I said, there's complex, legal, ethical, etc. There's a special status of medicine. And I'm going to talk specifically about data mining and pathology databases because that's where I've got some experience. A little while ago, we published, I'm going to put lots of stuff like this up there just to show you that even somebody who's a part-time researcher can publish stuff. Particularly in an area where there's so much data. So we looked really at some of the myths that are associated with data mining. And we said that big data is universally big. No, it's not. A lot of the data that I'm going to present to you, isn't all that big. Might only be a couple of thousand patients. You still have to have a research question, otherwise you don't know what you're looking for, you won't find it. You have to say what your model is. You have to consider sampling theory, so you still need to look at the dreaded p-values. But there's more valuable information you can find, and you can use observational data if you can get it in medicine to help support these relationships. But some classical methods are inadequate to deal with big data. So I'm going to talk now about specific uses of big data, or semi-big data, around the areas of quality control, reference intervals, relationships modelling, predicting outcomes, and detecting poor performance in laboratories. So I'm going to go into some detail now, but please don't fall asleep. It's really the overview that I want you to have about what you can sort of do with this stuff and how it's being applied now. I'm going to talk about quality control. Who here is a laboratory person? So you'll know that with every assay that you do, you run typically a couple of quality control samples, and on the basis of the performance of those quality control samples, that is, if you get the right answers for them, you'll stop the run or you'll let the run of results go. So that's what quality control is Typically the mean and the SD of the control samples are used to assess performance. But what has happened in some disciplines is they've actually used patient data. The problem with using these sort of standalone quality control samples is that you might run 1 or 2 a day. You might run 10,000 patients a day. So if there's a problem with the assay, by the time you found out at the end of the day that it didn't work, what are you going to do with all those results that are wrong, that have gone out? So QC, standard QC, really doesn't work in laboratories, in big laboratories anymore. What some areas have done for a long time is they've actually used patient data. So most patients, particularly depending on the population that you sample from, if you're sampling from GP practices, most patients are probably normal. So you can actually use the average of their results as a form of QC, rather than using specific QC samples. So that way every patient that you analyse, every one of those 10,000 patients, contributes to the QC. So theoretically you can detect error much earlier on, because if there's some sort of issue with the assay, and typical issues with the assay is that all the values shift up or shift down because of some reagent problem or calibration problem. So you can actually look at all your patient values, and if they shift up or shift down, there's probably a problem with the assay. That's the basis of average of normals. Some areas of pathology testing, notably haematology, have been using this technique for a long time. Sort of unfortunately, it's called Bull's algorithm, but anyway, it seems to be more valuable than that. So I'll talk now a little bit more about this QC strategy and the concept of using patients. This is Bull's algorithm, so these are just patient results, and you can see that over a long period of time, they're actually pretty stable. All the hemoglobins and all these other tests for all different patients are about the same. So it's quite a reliable test. Certainly some analytes behave better than others, and it's related to the relationship between the error in the method and the variation in the population. But there are a whole lot of analytes that actually this works for. For a long time, people have tried to move away from this concept of a couple of QC samples because the runs have become so big. And in fact, the problem with them is the definition of a run. What's a run? There was a time when you do a run of 100 samples, put a control at the start, control at the end, and assess the run on the basis of the QC samples. Now analysers go 24 hours a day, so there's no start and there's no end. And you run controls when the shift changes, or when something else happens, and you don't run a lot of controls. So the concept of a run has gone. So people have come up with rules like this, and I must admit I have problems trying to interpret that too. But basically what you're trying to identify is where's the risk to patients. So we want to move away from running QCs, which is a very artificial thing, to something about patient risk. There's an American who's done a lot of work in this, called Curtis Parvin. So it's all related to this issue about run length, and it's risk-based. Pathology is moving towards a risk base rather than compliance, and we can compare the probability that we detect error, probability of error detection, and that's the probability of rejecting a run that contains critically sized errors. He's been publishing on this stuff for some time. He's actually patented the method, and I just cannot understand it. So that's a problem with that model. Nobody can understand how it actually works. Some people came out, a couple of Spaniards, and they in fact, and you can see the key things there are quality control, risk, that's what it's all about, that's where we're heading. And they actually did a whole lot of nice graphs and found that in fact it tells you no more information than classical QC theory. So that's a problem. But the concept of using these patient averages of normals, that's what it's called, the average of normal patients, has been around for some time. Chemists have started taking up on it, and in 2015 a new model came out which seems to work. The sort of the variables that you use is you take a mean of patients. How many patients do you mean? 50, 100, 200? And some other things about how you deal with the results in that block. So that's not important. The main thing is this concept of average of normals. I've got some colleagues in Singapore where we've been trying to finesse this method. So this was looking at an average of normals, an average mean of patients, to see when the run fails, and we think we've come up with that. We've looked at, say, verification out of control situations by this same approach, and that seems to work. And there was a recent episode in South Australia, even hit the press, where a whole group of patients, male patients, who were post-prostatectomy patients, were being monitored for PSA. Normally in a male who's had his prostate taken out, there should be no PSA. So post-prostatectomy, they monitor PSA levels, and as soon as the PSA level starts to climb, post-prostatectomy, there's probably a secondary there. So they've been monitoring these patients. What the oncologists noticed was that they had a gut feeling that there were more patients coming through who were now becoming positive than they'd noticed in the past. They used to have a number like 40% of patients used to be positive for PSA post-prostatectomy. They found from the lab that it climbed up to 80%. So the oncologists trust the results. They phoned up the lab and said, I think there's something funny going on here. There's more positives than I used to see in the past. This is this concept of average of normals. You know, you can see if you're following a group of patients that something changes in the population. The lab said, no, everything's right. Our QC is great. There's no problems. But in fact, it turned out that the oncologist was right. There was all hell to play in the lab, but it just shows that in fact this gut feeling, this sense of patients, more patients being abnormal now than they were in the past, actually works as a form of QC. And what this tells you is trying to detect where sort of the abnormality may arise. There's still a lot of stats in it, so this is nothing magical. It's still based on statistics, but it's using patient samples rather than QC samples. And again, that's just to show you some idea of this sort of data, you know, how big is big data. We're using sort of 15,000 patients. And that's just to show you what it looks like. That's where you get this normal QC, sort of something rather fails beyond 2ST. And this is the paper about this concept of a number of positive patients. So once at some stage when the number of patients who become positive, who before weren't positive, is actually a QC process. And we believe we've shown that in certain conditions, it's just as accurate as QC, conventional QC. So this gut feeling that there's more people with brown hair in the room than there were yesterday actually works. And this is just some data to show that in fact it's more sensitive than conventional QC. There's always stats behind it, and the stats, there's nothing magical about the statistics. You know, this is just a block size, it's just the same sort of stats you've seen before. And the QC is exactly the same. So this is the QC chart for the number of abnormals, the number of outliers, so more outliers than there used to be before. And as well as looking at the average of normals, we've started to look at just not the average of normals, but the average of the SD of the population, because that tells us something too. No one's ever thought of that before. It's amazing. And that just tells you that you can use these different tools for different situations. But the key thing is we're not using QC samples, we're using patient samples. So this is real-time monitoring of QC processes using patient samples. And that might not sound much, but in an average laboratory, they'll probably produce a result every 10th of a second, and there'll be 10,000 patients a day and maybe 30 results every 10th of a second. So there's a lot of data coming out that in real time has to be averaged and etc. Other uses for patient data, reference intervals, you know, what's normal, what's considered to be normal, and biological variation and risk. I'll keep coming back to risk. That's the way we're focused these days. You can use, there's a hierarchy of what's good data and what's not so good data. Good data involves something to do with a clinical outcome. Not so good data involves something to do with biological variation. Not so good data involves something to do with expert opinion. So we all want to have clinical outcome data, but in fact in terms of clinical biochemistry, there's only one assay that we've got that has some relevance in terms of there's a clinical trial that's shown a relationship between a biochemical parameter and a disease outcome, and that's HbA1c. This is the technique that we use. Again, it's all statistical, based on Badrick, who was counting fish in the Bay of Bengal in 1963. And on the basis of this, that process, we've tried within Australia to in fact have the same reference intervals for common analytes across all of Australia. So we've started. A key person in this is Ken Sakaris, who works in a sonic lab, and he's looked at something like 800,000 patient results across Australia to come up with common reference intervals. And again, there's lots of information out there about it. So again, using pooled data from all parts of Australia to try and remove any sort of ethnicity, and also platform issues, instrument platform issues, to come up with common reference intervals for Australia. The Swedes did it some time ago, but Australia's doing it now. I've had a sort of an interest in really when are two results different. So if you get something assayed today in a lab and you get it assayed tomorrow or in 3 weeks' time in a lab, when are those two results actually different? That might sound sort of naive, but it's core to what we're trying to achieve. And it's sort of interesting. So a long time ago, I sort of developed this interest, and what sort of we found in those days, 2004, a majority of analytes are inappropriately reported when you take into account the analytical imprecision of the method. So the labs report results that really aren't that precise. Again, continue the theme a bit longer, taking into account measurement of uncertainty, which is really more errors than just analytical inaccuracy, but really two results. How different they are depends on the accuracy that you think there is when you measure them. And again, we did some reverse engineering, applied some statistics to that same problem, and looked at a whole lot of common analytes. And what we found was that this is a series of common analytes that are commonly measured. We wanted to estimate how many patients, assuming there's a Gaussian distribution, how many samples do you need to determine a reference interval, okay, using normal Gaussian statistics. And we found that in fact there's remarkable differences with some analytes. So for most tests you need 100 or thereabouts. With some tests you need 9,000. And you think that's a funny thing. But that's because the test units are over-reported. The number of significant figures in the test that's reported are too many. So for that particular analyte, we report, it's MCH, we report to the first decimal place. The accuracy of the method doesn't really allow us to do that. So rather than to report to 5.2 or 5.3 or 5.4, we should probably just report to 5. Or maybe 5 or 10 or 15. Okay. And the significance of that is that you can do all this sort of data analysis, but if in fact you truly can't tell the difference between two numbers, that's going to have an impact on what you're trying to achieve. Can you see that? So now we'll start to delve a bit further into things. So these are more specific things that I've been interested in. So in terms of a profile, I'm talking about, you go to the local doctor, they write something out, you go to have a blood test done, and 15 or 20 tests come back. So do you need that many tests to give you the same information? Look for relationships between them, which is sort of similar to that, using data to generate new models and looking at changes within patients and predicting outcomes. Okay, that's what we're going to talk about now. This was a sort of interesting study that came out from the UK with a liver function test. In Australia we typically have a 7-parameter liver function test, so we measure 7 things. In the UK they were going to measure 4. And interestingly enough, even though this is a sort of a research-based study, there was no data to support it. That's just what they felt like doing, experts rather than clinical data. So we did some work. This is some colleagues I got from ANU, we did some work just to see, in fact, are there redundant components of the profile? And all this shows that there are. So we came up with data based on a whole lot of data from where I used to work to show that, in fact, there's redundancy in a lot of the analytes. So rather than measuring 4, you only need to measure 2 most of the time. And similarly, we did the same thing with vitamin D in chronic disease. We sort of looked at vitamin D in chronic kidney disease, and we found that the 2 determinants of your renal function, your EGFR, the 2 determinants of EGFR on this data were age, that's no surprise, and vitamin D. I'll stop there because I don't know what it means. And I guess this is one of those problems where something pops up and you don't quite know what it means. So we don't know what it means, but there you go. We're going to do some more work and see if somebody else can tell us. I'll now talk about anemia. I've got a particular interest in anemia. I never used to be interested in haematology, but I crossed the Rubicon and now I love haematology. What percentage, this is a bit of trivia, but I think trivia is always good. What percentage of, you know what red cells are? I don't want to talk, but you know what red cells are? Yep, this one. How many, of all the cells in your body that are yours, not some bug, all the cells in your body, what percentage of those total cells are red cells? It's about 30%. So 30% of all your cells are red cells. Isn't that amazing? What's the other amazing thing about red cell? I think it's amazing. No nucleus. Isn't that incredible? So you would think something without a nucleus, you know, the whole idea of having a nucleus is that you can respond to external stimuli and make new proteins, lots of stuff. Red cells don't have that. How long do red cells last for?
3 months.
About 120 days. How does that work? They last longer than lots of other cells in your body without a nucleus. Aren't they amazing? I think red cells are amazing. So anyway, you know, with anemia, specifically, you know, the commonest causes are iron deficiency. How many people are iron deficient in Australia? I'll take that back. How many young females are iron deficient in Australia? Depends on the classification, but about 30%. Okay. This is just a normal blood film. There's those red cells. I think they're amazing. And the sorts of things that we normally look at are hemoglobin. We look at the red cells, size, colour, blah, blah, blah. And in a haematology analyser, this is important for later, in a haematology analyser, you measure sort of the mean size of all the red cells, but you also measure the SD of the differences. Okay? So why would you do that? I'll tell you. When you classify anemia, there's either little cells or big cells. So if this is a normal cell size cell, normal red cell, there's 2 sorts of anemias you can get that I'm going to tell you about. There's lots of others. You can get iron deficiency anemia. And iron deficiency anemia, you get smaller cells. Why is that? Do you think? What happens if you've got iron deficiency anemia? What does the iron do? Hemoglobin. What are red cells packed with? Hemoglobin. So therefore, if you don't have much hemoglobin, not surprisingly, the cells are smaller. That's okay. The other commonest form, particularly in older males, which I didn't know until we looked at this data, the commonest form of anemia in older males is vitamin B12 deficiency. What does vitamin B12 do? It's involved with making new DNA. It's in the sort of the one carbon transfer mechanism, and it's involved with making DNA. So with iron deficiency, the cells are smaller because there's not enough hemoglobin to pack in there, because there's not enough iron to make hemoglobin, so that's easy. Why, if you can't make DNA, which is what happens if you have insufficient B12, why are your cells bigger? I only found this out 10 years ago, so don't feel bad. What happens when a cell makes DNA, usually? This is long before they're released into the circulation. Makes DNA, splits the DNA, right? Cells go that way, you divide your cells, okay? And normally red cells divide about 12 times. Start off big, and they keep dividing and get smaller and smaller because they split off. If you haven't got any DNA, or you can't make DNA, you can't go through the 12 divisions. You only get to about 8. So you put out bigger cells. And that's interesting, but it's useful for later. So abnormal blood films, there can be sort of different levels of coloration, which is related to the immaturity of the hemoglobin. There can be funny shapes, or there can be funny shapes. So, and I said, this is key. What's anemia? What's anemia? Anemia is what somebody classifies it as, based on a ferritin, or an iron, or a hemoglobin. But they're often quite arbitrary, because really, you don't suddenly go from normal to anemic. It happens over a long period of time. So there's this whole range of values, like there is with most disease. There's a whole range where at some stage somebody says the cutoff is X. So you go from one day being not anemic to the next day being anemic, and you wouldn't know the difference. So what's being anemic mean? Something to do with your ferritin level, probably. And this is some work from Ken Sakaris, based again on 100,000 or so patients. And I just want you to look at the patterns, because I think they're lovely. This sort of relationship in young women between, this is hemoglobin, and their hemoglobin should be about 135 or thereabouts. And ferritin, at this stage, you're defined to be anemic by your ferritin level at about 15, and your ferritin gets to about 15. So you would be deemed to be anemic around about here, but you can in fact see that by the time you're anemic by your ferritin, you've actually lost quite a swag of hemoglobin. So it's all based on somebody's idea of a number. There's thought that in fact we should change the cutoff from 15 to 30, which looks a better number in terms of the drop-off in hemoglobin, but all of a sudden 50% of Australian females would become anemic. And what do you do with that? And this is just with elderly women. I shouldn't have said old. I'm one of those old people. But again, it's just these lovely relationships that they found by looking at all this data. It's only by looking at data that you can sometimes find interesting things. I had an interest in doing some stuff to try and come up with better tests that could be used, cheaper tests that could be used in developing countries, rather than expensive tests like vitamin B12, which are very hard and expensive to measure. And also I was interested in trying to understand a little bit more about hemoglobin, because I was amazed to find out how little we know about hemoglobin. Like, for instance, why the average male's hemoglobin is about 145, 150, average female's hemoglobin is about 135. How does that work? No. Even if they're fully iron supplete. You can feed them up if you wanted to, and the hemoglobin won't go above that. Why is that? So why is that? Does that mean you're all tired all the time, listless? Yes. Yeah. So it's sort of fascinating. And again, I've been involved with this stuff for a long time. I never thought about it. Who knows? Who really knows? But there are two other types of creatures that have the same relationship, where males and females have different levels of hemoglobin: fish and amphibians. So it's not just us. It's not related to hormones. So being a male with lots of blood is good, isn't it? Is it? No, it's not. No, males die younger. We've noticed that. It's a big concern of mine. It becomes a bigger concern the older I get. But males die younger. Why do males die younger? Mainly cardiovascular accidents caused by
smoking,
and
drinking,
and
stress,
yes, and
Marriage.
and
depression.
yes. Gee, I'm surprised I'm still here. What else? Sorry? Cholesterol. And what else?
Everything.
Hemoglobin. Hemoglobin is bad for you. Males would live longer if our hemoglobin was the same level as females. Honestly, one of the reasons why we have the cardiovascular, not the smoking, but one of the things why we have the higher blood pressure and the more cardiovascular accidents is because we're pumping a lot more blood around. It's thick, viscous stuff like a little heart. That's why we die. That's one of the reasons why we die sooner. So why have we got extra blood? What?
Faster.
Sorry?
You can run faster.
Probably. And what else?
Aspirin.
No. Why have males and why male fish and why male lizards got more blood? We're going back into prehistory here.
Reproduce?
No. I worry about some of the answers sometimes. Why have males got more blood? Think sort of primeval times when it might have been us against an amphibian. That's right, and we needed to have a little bit we could give away, or in case we got ripped to bits. So that's probably why. Pretty interesting. So how come?
Is it in spiders where the females are aggressive? Do they have more hemoglobin?
I don't know. No. I say I worry about some. That's, and the other thing that follows from that is that female red cells, there's no such thing. In females, their red cells give up oxygen more effectively than males. Otherwise, it's never going to work. So it's not just the fact we've got more hemoglobin than you have. We've got more females, sorry. We've got more hemoglobin because we might lose it when we have our next fight. But even without that, females are more effective at oxygenating their peripheral tissues. Isn't that fascinating? It's not hormone related. It's not hormone related. It's not dietary related. Now you can go away and look all that up. It's all about evolutionary biology, and it's fascinating. It's fascinating. But it's one of those things that I've never thought about. So we just looked at some data. This is from getting back to the anemia stuff. We looked at a whole lot of data, and we found with females that females have this sort of pattern of iron deficiency. No surprise there. We looked at males, and you just can't see it, but in fact we found the pattern was with B12 deficiency, which I was surprised about. Now I'm going to go back to RDW. So I'm building up to something here. I'm going back to RDW, which is the standard deviation of the red cell sizes. Okay. So what happens with your red cells is, say, they divide and they get smaller and smaller and smaller, and then they go to the spleen, and they sit in the spleen for a couple of days, and during that time they mature further, get a little bit smaller and smaller, and then they're released into the circulation. Or that's what we think. That's what we thought. So one of the consequences of that is that if your marrow is pushing out immature cells, they're bigger. If your marrow, and if you're iron deficient, if your marrow is pushing out iron deficient cells, they're smaller. So for a little period, at the period where you become iron deficient or you become B12 deficient, the SD of your red cells increases because you've got big cells and little cells as well as normal cells. So the value of the RDW is that probably you can pinpoint when somebody is about to become anemic. And we started to look at that because I've got an interest in RDW. I'm all passionate about RDW now. And in normal laboratories, lots of Australian laboratories don't even measure RDW because they think it's a waste of time. They use it maybe as a QC process, but they don't measure it because they don't think it's any use. Boy, were they wrong. So we started to look at, really, to try and detect this peak in RDW, so the point at which you become anemic, because this, I thought, would be useful in developing countries. So we looked at a whole lot of data in these patients from big private laboratory, and I'll just show you stuff because pictures are good. These are the sorts of patterns of RDW you can get, lots of patterns. This is the relationship between RDW and ferritin. So round about here, the 15, remember conventionally, that's where you become anemic. But all of a sudden, people started to find other things about RDW. We looked at RDW and looked at if there was any connection between RDW and other tests, and not surprisingly there was with iron and B12. That's no surprise because it's supposed to be a marker of those things, hemoglobin. We then started to look at individual people. So rather than just look at the whole population for hemoglobins for today or 6 months, we tried to track down individuals and followed individuals over a long period of time, sometimes up to 5 years. There's sort of ethical issues associated with that, but we did the right thing. And we found these patterns, all these sort of funny patterns. And I just show you that there's different patterns. It doesn't matter what they mean. We saw people whose RDWs were going up and down. This is the reference interval in the middle. Some people went down like this. Some people went up and down. There's all these sort of patterns. We didn't quite know what it meant. Remember, RDW was meant to be something that tells you when people are about to become anemic. And we plotted them all against time. With some of them, we could see that in fact what it was that people were becoming anemic, as that's what RDW is supposed to tell you. So as they became iron deficient, their RDWs sort of went up, down. And the question remained, why are these other patterns? What's RDW doing in these other patterns? There was a suggestion that it was something to do with inflammation. And round about this time, sort of round about 2013 and this time, all of a sudden there was all this data coming out on RDW. And it turned out that RDW appears to be predictive of death in a whole lot of diseases. Hepatitis, people in ICW, people with sepsis, people with lung disease. Just before they died, their RDW went up. Conventional wisdom was that RDW would take some weeks to change. They've actually found that people, just before they died, their RDW would suddenly shoot up. So that was interesting. Couldn't explain it, but it was interesting. And the other thing, therefore, that people started to think of, well, if RDW is useful in sort of potentially predicting when somebody's going to die, and I'm in ICW with all these patients who are pretty crook, I might use RDW as a way of indicating which ones might die. So, in fact, there are current ICW or ICU scoring processes where when you go to ICU, they score you using all sorts of techniques, Apaches and SAPs and all sorts of funny numbers like that, based on a whole lot of physiological data and pathology data. They give you a score. And the theory is that if your score is high, that's all right. If your score is low, that's not so good. And the theory is that if your score is low, maybe it's not worth spending a lot of time on you. But in fact, the score sort of turned round, and in fact those people who had abnormal scores, they worked harder at because they knew which ones to put all their attention into.
What time scales are you talking about here?
Physiological, hemoglobin, white cell count,
So
temperature.
when this changes
Yep.
before you die is 1 hour before you die?
No, I don't. I know you're excited, but just I'll tell you soon. No, it varies. It varies. So it can be a couple of hours before.
Because if you see media before, it's not very visible.
There's a range of results. So one of the things we looked at in this paper, we wanted to add RDW to the SAP score to see, or to Apache score to see if it made any difference. But also, just by the by, we also had a lot of other data that we mined with these patients. And we in fact found there was no relationship between RDW and CRP, so it's not inflammation. It's not inflammation. We added RDW to the Apache 3 this was, and it made a marginal improvement. You know, Apache's a pretty good score, so it made a marginal change, but it sort of proved the point. So round about now, I'm going to introduce this idea that you can learn things from doing data mining, learn things that you didn't expect to learn. So these people who are from the States mined a whole lot of insurance records of about 18 million patients in the States. And the insurance records have got a lot of information. And they mined this data looking at RDW and seeing if there was any relationship in their data. And their data is more complete than the data that I had access to, in that they've got a whole lot of medical and clinical history with these patients. And what they were able to support was a new theory for RDW, and that is that RDW is actually driven by release of erythropoietin. So when you're hypoxic, you get a little burst of erythropoietin, and that in fact releases these immature red cells that sit inside your spleen, or maybe even in the lining of your larger blood or your smaller blood vessels. So there was a theory that hadn't been really known before about this connection between RDW and erythropoietin. In fact, they looked at, so this is the sort of database that they used, 32 million, and it got down to 2.3 million with one RDW result, and they stayed there for a period of time. That's the sort of database tree that they used. So what that did, when we looked at our data again, is actually explain why we had the funny RDWs that we had. And the last thing I'll say, I think the last thing I say is detecting poor performance. So this is a completely different use of data mining itself. There's a new project that I'm just about to start, if I get some funding for it, and I'm hoping I'll get funding for it. We're actually going to look at laboratory data and see if we can look at gross laboratory data, external quality assurance data, that's what I do, and see if we can detect using that data any relationship between poor performance in that and a poorly performing laboratory when they're assessed by an independent assessor. So that's another use for laboratories. So it's going to be linked with NADA assessments. Hopefully we can pick up early markers of failure before the NADA assessors go around and detect it. There may be markers, we've done some preliminary work, and it may be related to particular analytes. So some labs that are not very good, there are some assays that they don't do very well, which is amazing. And these just people I work with. Thank you. Oh, that was... There are all sorts of seasonal variations. You know, our cholesterols go up in winter. There's daily variation with growth hormone and cortisol and a whole lot of things. Why does your cholesterol go up in winter? You don't go out and you eat more, believe it or not. And there's probably a physiological reason to build up a bit more fat when it's cold, so that's a well-known relationship. Also things like your CK, your creatinine kinase, which is related to muscle mass, they tend to go down in winter because you don't go outside. You know, vitamin D, no surprise there. Urea, which is related to protein breakdown, that can change in winter and summer depending on what you eat. So there are a lot of analytes that have all sorts of diurnal, monthly, seasonal variations. And summer, I say vitamin D levels sort of fluctuate, not surprisingly. Seem to. So you're right. In fact, hemochromatosis is worse than having iron deficiency. I'm not aware of, apart from sort of the penultimate phase, I'm not aware that there's a relationship between I've never seen anything about hemochromatosis and RDW. And the other thing, even now there's all this stuff about, I've been a laboratory person for a long time, a medical laboratory person for a long time, and I thought they knew something about hemoglobin. I thought they knew something about red cells. It's amazing how little they know. You know, like the average life of red cells in males and females is quite different. Average lifespan in a female is about 107 or 5 or something. In males, it's about 130 days. So that's interesting too. That's probably because you use yours more effective than we do. Just made that up. But there's all sorts of things. And this idea of 120 days, I think that was, and I'm almost exaggerating, but not much. It was based on about 15 people in 1930 or something. I don't know, but I have faith. I mean, I'm sure there was a time when, I just remember, as Andrew said, I'm sort of an amateur mathematician, and around about 1890, a guy called Hilbert came out, and I'm making this up, but I'm close. He came up with the 95 problems to solve, and then there were no more problems in mathematics left. So, and I think that lots of people have done the same thing. They've said, right, we've just got to solve this number of problems, and everything will be, that's it. There's no more human knowledge to find. So, but each time, we've gone further than that. We've developed the tools, so I have no doubt that there'll be a solution. I just can't quite get over. I've got a BSc in computer science as well. I did that when I first moved into laboratory. So I've got a specialist qualification in computer science. I actually did it before 1985, before personal computers were invented. So I've got a whole degree that's probably not worth anything. I can talk about assembler languages and I can talk about sort of big systems, but it was completely replaced sort of almost overnight. And I'm not that old, and I can't quite get over the fact that when I go on the train these days, everybody's just looking at their mobile phone. I can't quite get over the fact, because I'm old, that when I go out, there are people, couples at dinner, sort of doing this. So, and all that's happened in the last 15 years. So I have no doubt we'll solve that problem. And I think it was, wasn't it Watson in 1957 or something like that, who said we'll only need one or two computers in the world. I think it's one of those things, there's all this cause and effect, and it's obviously that the changes in the RDW reflect sort of changes in red cell size and other sort of mechanisms that are causing early release of immature cells or release of cells that are either large because of the lack of DNA or small because of the lack of hemoglobin. So RDW is sort of a summative parameter. And lots of those changes are because anemia causes lots of other things. So the hemoglobin goes down and the hematocrit goes down, all that sort of stuff. So it's sort of limited, and that's why most labs don't report it. But then it turned out that in fact RDW was first noticed to have some connection with mortality. And again, people didn't really know why. They thought it was inflammation or something to do with sepsis, but in fact that's probably wrong. And now we find that maybe it's a very indirect measure of erythropoietin. So I guess it's one of those tests that's been around a long time. We thought we understood it, and we didn't. And now maybe as a sort of a broad marker of a different disease process than what we originally used it for, it might be useful. It's like ESR. If you know about erythrocyte sedimentation ratios, it's one of those things where you can't actually work out why it's useful, but it is. And it represents a whole lot of changes in red cells and serum, but it's useful. There are two causes of small red cell anemias, microcytic anemias. One's iron deficiency, the other is thalassemia. In thalassemia, you don't get an increase in RDW. In iron deficiency, you do get an increase in RDW. So it was used to differentiate between iron deficiency and thalassemia. That was its original use. There's better ways to diagnose thalassemia now. That's why it went out of use.
That was a fascinating talk. I didn't mention that there is one other qualification that Tony has, and that is he's a visiting fellow at the Australian Institute of Health Innovation, so that means you might get to see him a bit more. And
I get lunch.
And you get lunch. Sometimes. Obviously your PhDs in quality management and mathematics are serving you well. Thanks for the presentation, and thank you for everyone for coming.
And thank you.